Hello students, this lecture is about the structure of eye. A pair of eyes are located in the sockets of skull called orbits. So here we are talking about the structure of the eye. So first, this is eyeball. The wall is made of three layers. We call external layer, middle layer, inner or internal layer. So first we start with an external layer. So just observe the diagram. There are different colors to represent different layers. So first is the outer layer. So the outer layer is actually made of dense connective tissue which is called as sclera. So this entire black layer from outside is the dense connective tissue layer which is called sclera, which is called the white of the eye. What you can see, the white color in the eye, that part is the sclera. So anteriorly the sclera continues as cornea. Cornea is a curved surface. The curved nature of the cornea acts as refracting structure. So it refracts the light. Next, always the cornea is kept moist and dust free with the help of tear glands. Now this is the external layer. These are two structures called sclera and cornea. Now external layer is without blood vessels. So cornea transplantation has no graft rejection. Reason is that there are no blood vessels. Next, the second is the middle layer. Now middle layer is actually called choroid coat. If suppose I say sclera is the outer, second is the choroid coat, which is the middle layer. So this is middle layer is choroid coat, which is actually pigmented with melanin as well as richly supplied with blood vessels. It's bluish in appearance. You can say the color is blue. That's a bluish in appearance, right? Now this choroid is 2/3 of this posterior part of the choroid is thin, whereas in the anterior end it becomes thick and forms ciliary body. So choroid forms ciliary body. In the anterior portion of it, it will become ciliary body. Now the ciliary body is with ciliary muscles. Next, ciliary body continue further as opaque and pigmented structure called iris. The green one is the iris. Iris is with muscles like ciliary body. Next, iris enclose an aperture. This aperture enclosed by the iris is called as the pupil of the eye. Now what is the function of this aperture? Now during bright light, the muscles of the iris, they constricts. As a result, the aperture or the pupil becomes small, so making less light to enter inside. In case of like dim light, the muscles of the iris relaxes. As a result of that, the pupil becomes like large, allowing more amount of light to enter inside. Next, eyeball also has a lens. Lens is held in its place with the help of suspensory ligaments. In these suspensory ligaments are connected to the ciliary body. Under normal conditions, these suspensory ligaments hold the lens under tension. So as a result, normal conditions, the lens is flat. So flat lens has more focal length, so that the light rays coming from a far object which are parallel can enter through this flat lens. Next, whenever the muscle, the ciliary muscles contract, the suspensory ligaments become loose. When the suspensory ligaments become loose, lens is more or less cylindrical. As a result, its focal length decreases, so that light rays coming from a near object, right, which are scattered, so they will enter through the lens. So this is how the lens adjusts its like size to accommodate the light rays from a far object or from a near object. That is how the lens works. Next, lens is actually a crystalline structure. It's a proteinaceous in nature, which is transparent. Next, like in the eye, this is your middle layer. Next, inner layer or internal layer has one pigmented epithelium and one retina. So this entire red one is the innermost layer, which is one is the outer is the pigmented epithelium and the entire red one is the retina. So to make it very clear, just look at into this diagram. So all these cells, this forms the pigmented epithelium of the inner layer. Next, below this is the retina. This entire part is retina. This entire part, which you can see here, the entire red structure is retina. Retina is in turn has 3 different types of cells. Actually, retina has 10 different layers, but whereas here we are talking about only 3 layers of cells. So one is the outer layer. Outer layer is towards the choroid. This is called receptor cell layer. Receptor cell layer, which consisting of photoreceptor cells. Next, below this is the bipolar layer. Bipolar cells are present, and inside is a ganglionic layer. It's a ganglionic layer. Just observe this diagram. So first, this part, this all the red cells part is called as photoreceptor cells. These are photoreceptor cells. These photoreceptor cells are of 2 types. They are rods, which are rod shaped. These are rods, and which are cone shaped. You just see the diagram. These are cones. Rods and cones are the 2 types of photoreceptor cells present just below the pigmented epithelium, which is a layer present in the retina. Now, rods are made of a protein called rhodopsin, right, which is called as visual purple. Next, rods are responsible for twilight or scotopic vision. Scotopic meaning is the vision during dim light. Twilight is the period before sunrise, after sunset. That is called as twilight. So rods are more in number, and rods are responsible for vision, twilight or scotopic vision. Whereas your cone cells are less in number compared to rods. Cones are 3 different types of cones are present with the different photopigments in them, which are sensitive for colors like blue, green and red. So these different, like photopigments, when they receive a particular amount of light with the different wavelengths, we can see different combinations of colors. Like when all these 3 types of cones are equally stimulated, we get a sensation of white light. So cones are for color vision, rods are for dim light or twilight vision. Next, these are the photoreceptor cell layer. This is the red one. Imagine this red one is the complete photoreceptor cell layer, which is having rods as well as cones. This is P, I am making it photoreceptor. Just below it is a bipolar cell layer. So this is your bipolar cells. So imagine in this diagram, this is a bipolar cell layer. All these are bipolar cells. All these are bipolar cells. So on both sides, this is bipolar cells. Next, just below the bipolar is the ganglionic cell. This is the ganglionic cell. This is the ganglionic cell. So when you talk about retina, retina has photoreceptors to outside and ganglion to inside. This is the inside portion. Ganglion cells are to the inside. Now all these ganglionic cells, from every ganglionic cell arises a nerve fiber. All these nerve fibers fuse and form an optic nerve, which leaves the posterior pole of the eyeball, from where retinal vessels enter, retinal blood vessels enter the eyeball. So this part of the eyeball where there are no receptor cells, so this is not sensitive for light, so this part is called as blind spot. Next, medial to this is a yellow colored structure which is called macula lutea, and this macula lutea has a central pit which is called fovea centralis. Fovea centralis is devoid of rods, it has only cones, it has highest resolution. Next, so this is what is this innermost layer, these 3 layers. Along with this, there are some chambers. So like the space between the cornea and the lens, this part is called as aqueous chamber. This is aqueous chamber. So between the lens and the cornea, this is called aqueous chamber. This aqueous chamber is filled with aqueous humor. And what is it composed of? Aqueous humor is composed of glucose, protein like albumins, and there are some antibodies called IgG type of antibodies are also present in the aqueous humor. And what for it? The function of this aqueous humor is to provide nutrition to the cornea as well as to the lens. Next, the space between the lens and the entire retina, this is called vitreous chamber. This is vitreous chamber, and this vitreous chamber is also filled with the fluid called vitreous fluid. Now this vitreous fluid is 98% of this is filled with water, and also it contains a trace amounts of hyaluronic acid and it contains collagen protein. So this is completely a gelatinous part and it provides nutrition to the retina. So both aqueous humor and vitreous humor are formed from the ciliary body. So these are the different layers present in the eyeball. Now in the last we see the mechanism of vision. Fine. What are the light rays that enter the eye? Right? All these light rays, first they hit the cornea. So cornea allows the light to enter into the pupil, that is aperture, from there to the lens. And now lens focuses the light rays on the retina. Now here the point what you have to remember is when light falls, when light enters into the retina, light travels through ganglionic cell, but light will be received by your receptor cells. So light is traveling like this, but it will be received by your receptor cells. Now when light falls on your photoreceptor cells, like imagine those photoreceptor cells are, for example, rods. When light falls on rods, as we have discussed that rods contain a protein called rhodopsin. Rods contain a protein called rhodopsin. When light falls on rods, this rhodopsin splits into protein called opsin plus retinal. Retinal is an aldehyde of vitamin A. Whenever it splits, there will be some conformational changes in the rods, and those conformational changes will produce potential differences in rods. Now these potential differences in the rods are sent to bipolar cells. So this is a bipolar cell. So in these bipolar cells, these rods will make, potential differences are created in the rods, and those potential differences are sent to the bipolar cells. In the bipolar cells also potential differences are generated. Now these are called graded potentials. Now from bipolar cells, now these potential differences or graded potentials are sent to the ganglionic cells. Now in the ganglionic cells, action potentials are generated. Now these action potentials generated in the ganglionic cells are carried by the optic nerve fiber to the visual cortex area of the brain, where with the previous memory, that image is recognized. So this is how vision occurs.